Understanding CRISPR-Cas. Cluster regularly interspaced short palindromic repeats CRISPR-associated protein. Pause text and illustrations for detailed study. Refer to the previously posted constituent factors observed in viral diseases, parts 1 through 5. Virus spike protein structure and life cycle illustrations. Spike protein virus genetics and mutation. Characteristics of novel viral genetic variants possessing envelope spike proteins, constituent factors observed in messenger RNA vaccination, and messenger RNA vaccination immunity and pathogenic priming for background data related to this presentation. Clustered regularly interspaced short palindromic repeats, CRISPR, is a family of repeated DNA sequences in the genomes of prokaryotic organisms. CRISPR are derivations of DNA fragments that had infected the prokaryote. These sequences are elements of a prokaryotic adaptive immune system functioning as a type of acquired immunity for bacteria. They recognize and destroy DNA fragments of bacteriophages upon subsequent infection, functioning in the prokaryotic antiviral defense system, providing for acquired immunity. CRISPR prokaryotic antiviral defense mechanism illustration. CRISPR cascade protein bound to CRISPR RNA and phage DNA illustration. CRISPR-associated proteins, Cas, function as the enzymatic mechanism by which the acquisition of new spacers, as well as pathogen targeting, results from previous exposure to invading elements. An example is the Cas9 enzyme guided by CRISPR sequences that recognizes and cuts specific DNA strands complementary to the sequence. The CRISPR sequences, in concert with the Cas9 enzyme, function in genetic sequence editing in organisms. Cas proteins are numerically suffixed and classified, related to the specific function and pathogen involved. The CRISPR-Cas9 system involves the Cas9 enzyme functioning as a molecular scissor that cleaves to DNA strands at a specific genomic locus for the addition or removal of DNA fragments, as well as a piece of genetically predetermined guide RNA, gRNA, that is part of a longer RNA sequence binding the scaffold to DNA for Cas9 mediated cutting and binding at both strands of the DNA at the correct gene locus. Cas9, along with the gRNA, reaches the same locus in the DNA sequence, cutting across the double strands of DNA. gRNA locates and binds with specificity. Its bases are complementary and bind to the target DNA. The Cas9 nuclease possesses two active cutting sites for each strand of the double helix DNA. It unwinds the foreign DNA, checking for its complementarity to the 20 base pair spacer region of gRNA for cleavage to occur. The cell recognizes the damaged DNA and initiates the process of repair. CRISPR-Cas system proteins functions to change genetic memory by means of the spacers from viruses and plasmids, resulting in adaptive immunity against mobile genetic elements. Simplified diagram of a CRISPR locus. CRISPR-Cas9 illustration. The initial stage of the immune response upon microbial invasion by a bacteriophage is to detain and insert phage DNA as a spacer into the CRISPR locus. CRISPR system-derived immunity integrates short viral sequences at the cell's CRISPR locus, enabling it to recognize, remember, and clear infections. The CRISPR-Cas9 system is an adaptation of the naturally occurring bacterial genome editing system. CRISPR-Cas inhibits bacteriophage infection, conjugation, and natural transformation via the degradation of foreign nucleic acids that enter the cell. The CRISPR-Cas9 system can cleave DNA, as well as turn gene expression on and off. CRISPR-Cas9 as a molecular tool introduces targeted double-strand DNA breaks. CRISPR-Cas9 genetic editing process. In the gene editing tool CRISPR, a small strand of RNA identifies a specific chunk of DNA. Then the enzyme Cas9 swoops in and cuts the double-stranded DNA in two places, removing the specific chunk. CRISPR-Cas9 illustration. The CRISPR system has two components joined together: a finely tuned targeting device, a small strand of RNA programmed to look for a specific DNA sequence, and a strong cutting device, an enzyme called Cas9 that can cut through a double strand of DNA. Once inside a cell, the CRISPR system locates the DNA it is programmed to find. The CRISPR-seeking device recognizes and binds to the target DNA. Cas9 enzyme cuts both strands of the DNA. Researchers can insert into the cell new sections of DNA. The cell automatically incorporates the new DNA into the gap when it repairs the broken DNA. The CRISPR genetic locus provides bacteria with a defense mechanism to protect them from repeated phage infections. CRISPR has many possible uses, including insert a new gene so the organism produces useful medicines, help treat genetic diseases, create tailor-made organisms to study human diseases, and help produce replacements for damaged or diseased tissues and organs. Schematic flowchart of molecular detection methods for COVID-19 virus. CRISPR-Cas based platforms are being considered for the detection and inactivation of SARS-CoV-2. Milestones of development and application of CRISPR-Cas systems. New presentations coming soon.